Today we'll be doing a quick review of cell structure and function. So first things first, those cell models that you've always seen, they are not real. They exist as an idealized form of a cell. Not all cells are the same. Not all cells are the same shape, size. They don't even have the same organelles. So the basic idea that all cells are exactly the same, flawed. Red blood cells, white blood cells, and platelets, for example, are extremely different, even though they all come from the same source, the hematopoietic stem cells of your bone marrow. Red blood cells don't even have a nucleus, so they barely count as eukaryotic. They come from a eukaryotic source, so we still consider them eukaryotic, but they do not go through mitosis. They do not divide. So it's really just a good example of why all cells are different, or at least how they can be different. One of the biggest limiters to how big a cell can get is surface area to volume ratio. Surface area increases on the square, whereas volume increases cubically. So this is a square-cube relationship. As a cell gets larger and larger and larger, the surface area, the area needed for the transport of materials and the export of waste, gets less and less and less. So as organisms began to get more complicated and to evolve the traits that we know now, some cells went sort of folding, branching in order to get the size and shape that they needed. Other cells went multicellular, and that is a major differentiator. The other major limiter to how big a cell can get is the so-called genome to volume ratio. Because your genome includes all of the instructions needed to make proteins and all of the various mRNAs and so on needed to produce all of the functionality of the cell, if you have a cell that is too big for you to be able to produce enough of these controlling mechanisms, the cell will also die. So SA to V and G to V ratios are both limiters to how big a cell can get. When you actually look at prokaryotic cells, they don't really have much in the way of organelles, but they do have organelles. So the idea to say that they don't is false. What they don't have is membrane-bound organelles. So you have the bacterial chromosome, which is found in the nucleoid region, which is a large circular stretch of DNA. They could also have smaller circular pieces of DNA plasmids that are very useful in biotechnology. You have flagella that allows for movement. The flagella of a bacteria is made of flagellin, and it is a convergent evolutionary trait. Our cells, animal cells, can have flagella as well, but they are structurally different, even though they are functionally the same. The capsule and the cell wall work to protect the cell. The capsule prevents dehydration, it helps evade the immune system, whereas the cell wall provides structural support. The plasma membrane, just like ours, controls what comes in and out, allows for diffusion and transport. Ribosomes make proteins, and the fimbriae allow sort of gripping and holding of bacteria onto the host organism or whatever it's living in at the time. Animal cells and plant cells, these models that you're shown here are incredibly idealized. You might have hundreds of mitochondria, for example, not the 3 or 4 that are presented here. So we're going to go through each of the major organelles, but we do need to know the major differences. Animal cells have one organelle that plant cells do not, and that is the centrosome. The centrosome is made of 2 centrioles and is a major controller of mitotic events, and we'll get there in a little bit. The only other major thing that animal cells have than plant cells is just that we have a significantly more developed extracellular matrix than plant cells. But it kind of makes sense when you think about animal cells are motile and plant cells are not. So that intracellular communication is significantly more important in an animal cell. Now, plant cells obviously have cell walls made of cellulose. Those cell walls have openings called plasmadesmata. Those are both structures that we wouldn't have. The chloroplast, the site of photosynthesis, is a representative of the plastids, which is a major group of organelles that occur in plants that do not occur in animal cells. The only other major difference is the large central vacuole. It's not to say that animal cells don't have vacuoles, but we don't have this big of a vacuole, this large central structure, which is incredibly important for water control and transport. The nucleus is the most important of the organelles as far as DNA is concerned. It's made of 2 layers of membrane, the inner and the outer membrane, with a layer of cytoskeleton in between called the nuclear lamina. It has nuclear pores that allow material to come in and out, which is absolutely essential as DNA controls the blueprints, but it doesn't produce anything. The ribosomes in the cytoplasm do. Inside the nucleus, you have the diffuse DNA, euchromatin and heterochromatin. The euchromatin is open and available for use, and the heterochromatin is not. It's condensed and unavailable, and that makes a major difference between cells. A very dense region of euchromatin, where all of the genes needed for ribosome production can be found as the nucleolus. Some cells will have 1, 2, 3 nucleoli, depending on how many ribosomes they need. And it's not that it's a separate organelle, it's just all of the euchromatin needed to produce it is found in one place. Ribosomes make proteins. Okay, prokaryotic cells have smaller ribosomes than eukaryotic cells, but the function is the same. Bound ribosomes are attached to membrane of some kind, and the material that they produce are bound to leave the cell. Maybe just the cell, maybe into a different organelle, but they're going to leave the cytoplasm at least. Free ribosomes are made from ribosomes that are free ribosomes produce material that stays in the cytoplasm where the free ribosomes can be found. Now, the endoplasmic reticulum, there are many different types of endoplasmic reticulum. You're expected to know two right now, the smooth and the rough ER. The smooth ER's major job is lipid biosynthesis, as well as the function of detoxification. And from that process, it also makes the organelle, the peroxisome, which also detoxifies. The rough ER is studded with ribosomes, and that works with the ribosomes to produce the protein components of cell membranes. So together, the ER is a membrane factory producing all of the materials and molecules needed to produce more membrane. That bubbles off in the form of transport vesicles, which can be used in a variety of methods. Now, if that transport vesicle is holding proteins that are being released by the endomembrane system, it will then fuse with the Golgi apparatus. The Golgi apparatus is like the shipping and receiving section, modifying and labeling things for their final destination. So the Golgi apparatus uses those SRP, or the signal recognition proteins, to control where amino acids and proteins that are produced go. So this is that mail center sending things out to the cell. That might fuse with the cell membrane, or it might go to the nucleus or to another organelle. So the Golgi body also makes the organelle the lysosome, which we'll look at in just a moment. So the lysosome is a vesicle made by a combination of the ER and the Golgi body, full of hydrolytic enzymes. Those hydrolytic enzymes will allow digestion, hydrolysis, and those materials will recycle. So lysosomes are kind of like the cellular stomachs, breaking the material down to be recycled. Peroxisomes, made primarily by the smooth ER, are full of molecules that are capable of producing hydrogen peroxide. So this is going to have a combination of catabolic effects, biosynthetic effects. It's necessary for photorespiration, but it also is a major detoxifier. It makes sense that the organelle that detoxifies helps produce the other organelle that detoxifies. Vacuoles and vesicles are bubbles of membrane that store and move things around. Vacuoles are just larger. They're typically the storage ones, whereas vesicles are typically the transport ones, but they're all just bubbles of membranes doing different jobs. The mitochondria is an endosymbiotic organelle that produces primarily ATP. It is a double membraned organelle, so that shows it's one piece of evidence that this was endosymbiotically gained as a separate prokaryote. It has its own DNA that is in rings like a prokaryote. It has its own prokaryotic ribosomes. It moves by itself. It divides by binary fission by itself. And so the mitochondria is a very important organelle. The chloroplast is the main endosymbiotic organelle for photosynthesis. The surface area there from the thylakoid allows for a great deal of energy to be captured. It is a double membraned organelle, just like the mitochondria, with its own DNA, with its own ribosomes, with its own division. So it is also endosymbiosis is the major source of this organelle. Microtubules are the largest of the cytoskeleton components, and they work with motor proteins to move things around the cell. The MTOC, or the microtubule organizing center, is the major organelle responsible for producing these microtubules. So these are temporary transport structures, like roads that are built to move things around. For animal cells, we have our centrosome, which does this. For plant cells, the nuclear membrane does it. But those microtubules make up these movement structures, as well as the more permanent structures for movement, like the cilia and the flagella, are made of microtubules and motor proteins working together to move the cells around. And remember, flagella is needed for movement. So even though bacterial cells have flagella and eukaryotic cells have flagella, this is not a common ancestry thing. It is convergent. The movement of cells is an incredibly useful structure, so it makes sense that it's been selected for biologically. Microfilaments are the smallest of the filaments. These exist in a variety of places, like your muscles, for example, are made of microfilaments. They're very good at dealing with tension and pressure. And then it's this intertwined actin. So actin, you might remember from the actin ring of mitosis, a wide variety of functions there. The intermediate filaments are kind of like coiled wire. They're very, very strong. They're very good at bearing tension. The nuclear lamina is made of this. Your organelles are anchored into the cell's position through this. So this is the closest you get to an actual sort of permanent skeleton structure for the cell as far as cytoskeleton is concerned. The cell wall is made of a variety of materials, mainly polysaccharides, and which polysaccharide it is depends on what type of cell you are looking at. Cellulose in plants, fungi have chitin, bacteria have peptidoglycan, but it is a structural thing. Now, it's not an actual wall, it's more like a mesh, and you can see for an example here that you have a variety of fungi growing through these cell walls, or at least around the cell walls, connecting the cells. And that's one way how, like mycorrhizae, for example, that symbiotic fungus that extends the structure of roots and allows for plants to absorb way more water, directly transports that water in using these cell wall penetrating structures, these hyphae. The plasma membrane, of course, is extremely important, controlling the inside and the outside, made from a variety of phospholipids and proteins and sugars and cholesterols, and holding the structure in place. And then it connects to a series of proteins and sugars, proteoglycans, protein, glyco, sugar, allowing for cell-to-cell communication and recognition. Blood type, for example, comes from these marker proteins and sugars on the outside of the cell. Cellular junctions allow for either connection in the closed junctions or passive movement of material in these open junctions. In open junctions, you have the gap junction if you don't have a cell wall, and you have plasmodesmata if you do have a cell wall. These are going to allow ions and small molecules to diffuse, and it's very common in tissues and organs that work together. The closed junctions, you have the desmosome, which are like strong rivets of screws made of cytoskeleton, and then you have this webbing of cytoskeleton fibers that form the tight junction. So the gap junctions allow movement, whereas the closed junctions allow for solidity and connection. The desmosome creates strong points of bonding, where the tight junctions makes waterproofing possible. So that cell membrane is extremely important. We're going to look at how it affects transport as we go, but the cell membrane is a fluid mosaic membrane. If you took two cells and put them together and you came back to them over time, you would see that the organelles diffuse and move around. So these organelles are, these phospholipids are constantly flowing and moving and flipping around, and you'll see major differences. Now, the more saturated the tails are, the more solid it will be, or you could add cholesterol to fill in the gaps, and that would also decrease the fluidity. So temperatures, for example, low temperatures, cholesterol might be used to hinder solidification and hinder freezing, keeping those plant cells or whatever alive in winter. Or you could alter the type of fatty acid that makes up your phospholipid. But regardless, the fluid mosaic model is the best model we have today. We do see that the hydrophobic interaction of those tails is the weakest part, so you can freeze fracture and break this open to get an idea of what's going on. But you have these integral proteins that go all the way through, and you have the peripheral proteins that are facing one side or the other. But the inside and the outside of the cell's membrane are very, very different. Why? Because they have very, very different jobs. But where did all this stuff on the outside come from? It had to come from the inside. There's nothing that can make things on the outside. So that movement of vesicles and vacuoles, and we'll look at as the bulk transport and endocytosis and exocytosis, those are absolutely essential for the building of membrane. So when the vesicle fuses with the outer wall, that exocytosis, the inside of the vesicle becomes the outside of the cell membrane. So all of those receptors and proteins and things all come from endoplasmic reticulum produced material and modified by the Golgi for that endomembrane system interaction. That membrane, though, is selectively permeable. So things that are small and hydrophobic can diffuse through with no help. So oxygen, CO2, fine. But something like water that is hydrophilic, obviously, is small enough that it can get through in a limited amount, but not hydrophobic, so it's going to have a hard time getting through those fatty acid tails. Something that is large or charged is not going to get through by diffusion or passive processes, and active processes will be needed. So membrane proteins are a major component of this transport, allowing for cell-to-cell recognition and enzyme recognition and joining. So this is going to work with the ECM and work with signaling molecules. So membrane proteins are extremely important to this. But cell transport is the key thing right now. Material that can diffuse by itself, that is passive transport, and that's going to allow for free flow of material from high concentrations to low concentrations. If proteins are involved in moving from high to low without the expenditure of energy, we would call that facilitated diffusion. That could be a channel or some sort of pump, some sort of passive pump process. If energy is needed, ATP, redox energy, photon energy, whatever, that typically means you're going from low to high concentration, you're going against the flow, and that is going to require energy to do so, and proteins will always be involved. So diffusion happens before osmosis. You look at whatever molecules you are dealing with and you will diffuse all of them first. Then you will sum the molarity of both sides of whatever you're looking at, and then you will do osmosis. Osmosis is the diffusion of water. The water goes from where the water is high to where the water is low. So if you're looking at the solid materials like you did in diffusion, it's going to look backwards, because wherever there is more solutes, there's going to be less solvent. So wherever there's more stuff, there's going to be less water. So diffusion and osmosis look like they go in different directions, but they don't. They both go from high to low. It just depends upon what you are looking at. Transport in plants depends upon this idea of that water potential. And while there are a variety of components of water potential, solute potential and pressure potential are the only ones you need to care about. The solute potential is provided to you on your formula sheet as negative ICRT. I is the ionization constant. If the molecule is ionically bonded, the answer is likely to be 2. If it is covalently bonded, the answer is likely to be 1. If they give you any form of sugar, it's 1, salt, 2. If they give you something else, they'll have to give you some more information. C is the concentration, the molarity. R is the constant that is provided to you on the formula sheet, and T is temperature in Kelvin, which is provided to you on your formula sheet. But transport in plants depends upon that water potential, and we see the movement of plants. Pressure comes from the turgor pressure of the cell wall pushing back. So a flaccid plant cell, for example, even though it has a cell wall, will not have any pressure potential. And an animal cell, unless it's in some sort of pressurized environment, which I've never actually seen, will also not have pressure potential. So the solute potential is all you need. But that balance of water is tonicity, that water balance, that osmoregulation process. So a cell that is hypertonic will have more stuff in it than the solution that it's in. So that solution is hypotonic. That hypotonic has more water, so the water will go into the cell, causing it to swell and maybe lyse. Now plant cells like this. They like the water pushing in because they can push back with the cell wall and prevent from bursting and be turgid, which is that crisp feeling that is very good for plant life. Isotonic solution, the water is going in and out equally. It's not stable, but it is a dynamic equilibrium moving in and out, and that is the best situation for an animal cell. Plant cells, this is flaccid. It's survivable, but it's not great. Nothing likes a hypertonic solution, so like a very salty water like the Dead Sea. Everything is dead because the animal cells will shrivel or crenate, and the plasmolized plant cells, the cell wall is ripped away from the cell membrane as the cell membrane shrinks and the cell wall can't, and that is not good for anything. So the Dead Sea is dead because it's so salty. It's so high in salt concentration that it's hypertonic to basically everything. Active transport requires energy expenditure. So the ATP spending is the most common example here, and you can see an example of this in the sodium-potassium pump, which is a major component of your nervous system. That direct expenditure of ATP is a primary transport. So the proton pump on the bottom right is an example of this active transport. This example is a proton pump, so it's pumping protons from low to high, which is definitely a sign of active transport, so that we know that's going to be altering the pH. It's going to be more acidic outside. That difference creates a charge. That charge can be harnessed to pump other things back in, and that's secondary active transport. This is active transport that occurs due to the indirect expenditure of energy, which is a fundamental difference. Vesicular transport is for the large molecules. So you're going to have endocytosis, which brings materials in, and exocytosis, which brings materials out. The endocytosis comes in three major types: phagocytosis, which is for larger or solid materials bringing it in. This is extremely rare. Nothing with the cell wall can do this. You have to have a lot of flexibility in your cell membrane. So the white blood cell of animal cells is the greatest example I have of this, phagocytes, because obviously the name shows that they are very commonly seen doing this. Penocytosis, all cells are capable of penocytosis. This is small little bubbles. You can even do it through the gaps in a cell wall. So this is mainly looking at liquids and dissolved solute material. And then you have receptor-mediated endocytosis. So this is looking at very specific molecules that can be brought in based upon binding to a receptor. And this also is rare. Only certain specialized cells are going to be capable of doing this. So endocytosis, there's three major types, but exocytosis, there is only one. Now, where did the actual first cell come from? Well, there's many different theories, there's many different ideas. The most important one that we need to remember is the Operin and Haldane hypothesis, which basically is spontaneous abiotic biosynthesis. Years later, two scientists, Miller and Urey, actually tested their idea using atmosphere that they believed the early Earth had, which was a reducing atmosphere rather than the current oxidizing atmosphere that we have now. Oxidation is very harmful for the building of complex bonds. Think rusting. It's not great for biological structures either, those free radical oxygens. So in a reducing atmosphere, life, the building, these complexifying forces would be much more likely to happen. And so Miller and Urey showed that if the early Earth's atmosphere was like what we thought it was, we would be able to produce these abiotic molecules. They tried it again with a better understanding of what the gases were, and while it wasn't as successful as Miller and Urey, it did work. So there is definitely some evidence supporting this idea. Regardless, once you have biological macromolecules, the next major process is to create some sort of compartment. That compartment could have been something like a clay, like this montmorillonite clay, which is common in the Sidney Fox idea of the origin of life. Or it could have been protobionts, coacervates, some sort of protocell. That would also work. But once you have those boundaries, those separations, the next major step would be, is it genetics? Is it metabolism? We don't know. But we do know that once you have compartments, complex life began to occur fairly rapidly. Probably some component of both of them are likely, but the major step up was obviously cell respiration, which allowed for cells to have much more energy, and then photosynthesis. Eventually photosynthetic bacteria came about, and that drastically shaped the future of everything on planet Earth. And we know exactly when that happened because that's when you start seeing rust in the rocks. So this combination of events led to this major change in cells, but the energy components were arguably the most important. So most of those energy cells were evolved by bacteria. The early ancestral aerobic bacterium and photosynthetic bacterium were engulfed through the process of endosymbiosis, becoming part of the eukaryotic cell. So no, prokaryotes don't have membrane-bound organelles, but prokaryotes that are inside other cells can become membrane-bound organelles for that cell. So the mitochondria is an endosymbiotic organelle that our ancestral cells took in. Plants' ancestral cells went on and did that again and again with secondary endosymbiosis, and that's where the larger family of plastid organelles came from. Chromoplasts storing pigments, amyloplasts storing starch, elioplasts storing lipids, proteinoplasts storing proteins. So endosymbiosis is extremely important to that energy foundation that made complex life possible. But once energy was available, multicellularity became a fact. It was an inevitable conclusion to these processes due to the surface area to volume and genome to volume area limitations on cell size. The early idea was that multicellularity evolved once, and all multicellular life came from that multicellular life. But if you look back, it's not true. Multicellular life is a parsimonious process. It did not evolve once, it evolved many times in many different branches of life, all of which come together to make up the life that we think of now. So multicellularity is a great example of when simple sort of maximum parsimony is not always going to be true. The way things became multicellular was drastically different from type to type.